Today, we will be discussing a revolutionary gene editing tool known as CRISPR-Cas9. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This may sound complex, however it's quite easy to understand once you know the roots of CRISPR. CRISPR originated from E. coli. It was discovered that the DNA of E. coli contained these palindromes, segments of DNA that are identically read back and forth on both strands, were placed at regular intervals. Between these palindromic repeats were spacer DNA, the CRISPR DNA. The spacer DNA broke the palindromic pattern found in DNA by seeing properties. Why did this happen? It was discovered that the spacer DNA was actually DNA from bacteriophages to understand why it's there, by exploiting the next part of CRISPR, the Cas9. Cas9 is an enzyme that makes Cas proteins, which are helicases acting as DNA zippers, and nucleases acting as DNA scissors. When a bacteriophage attacks the bacteria, the Cas gene goes into action and saves the day. It does this by making CRISPR RNA from the spacer DNA that will combat the attacking bacteriophage by breaking it apart. In cases where there is no spacer DNA for a new attacker, the Cas protein will take the new DNA in, break it, and inject it into the palindrome, forming new spacer DNA. Basically, there are old infections saved in the DNA of E. coli. This may sound extraordinary, but we humans have those inner immune systems too, which is why we get vaccines in the first place. For us, we undergo a process known as phagocytosis, in which the pathogen, the attacker, is absorbed by white blood cells. Since all this happens in live cells, scientists have found a way to borrow CRISPR from bacteria and use it in humans. CRISPR is used in humans by switching genes on and off, and even adding in new ones. It can even edit DNA to the very letter. The importance of this is seen in cases such as sickle cell anemia, where the problem is traced to a single wrong letter. CRISPR-modified DNA simply snips out that letter and adds a new one. As you can see, CRISPR-Cas9 is a very promising tool. If something has this much potential, there's also a lot of controversy surrounding it, just like the public outcry in regard to GMOs. Given enough time and experimentation, CRISPR-Cas9 will cause a biological revolution needed to completely reshape the world we live in for the better.